Greetings and welcome to the United States Transhumanist Party Virtual Enlightenment Salon. My name is Gennady Stoliarov II, and I am the chairman of the U.S. Transhumanist Party. Here we hold conversations with some of the world's leading thinkers in longevity, science, technology, philosophy, and politics. Like the philosophers of the Age of Enlightenment, we aim to connect every field of human endeavor and arrive at new insights to achieve longer lives, greater rationality, and the progress of our civilization. Greetings, ladies and gentlemen, and welcome to our U.S. Transhumanist Party Virtual Enlightenment Salon of October 22nd, 2023. I am pleased to offer you today a discussion of how machine learning can be harnessed to advance drug discovery, particularly in the field of longevity, where, of course, it is of paramount interest to us to personally reach longevity escape velocity, yet there are so many options to consider and investigate. So how can this process be accelerated? How can we determine which targets are actually worthwhile to investigate and focus on? And this is where artificial intelligence and machine learning can become extremely helpful. So joining us today is our distinguished panel of U.S. Transhumanist Party officers and members, including our Director of Visual Art, Art Ramon Garcia, our Director of Longevity Outreach, Ben Ballweg, our 2024 U.S. Vice Presidential Candidate and Director of Citizen and Community Science, Daniel Tweed, our member, Alan Crowley, and our special guest today is Dr. Ivan de Weber. He is a PhD, a scientist, and entrepreneur behind pioneering concepts in aging genetics and stem cells. He turned to pharmaceutical bioinformatics amid an explosion in biomedical data. His quest for faster drug development led to the creation of Cortex Discovery, his company, which uses machine learning to accelerate the search for new medicines to cure disease and expand human longevity. So thank you, Ivan, and welcome. Please tell us a bit more about yourself and the work of Cortex Discovery. And I know you have quite an intriguing presentation in store for us.
Okay, great. So hello, guys. Maybe I'm going to start by the beginning. I think one of my first memories was my parents telling me that people had died and some of their friends. So I was wondering, and I was asking them questions like, do we have to die? Everyone has to die, but not us. But they said, no, we have to die as well. And I think that was a big. It was really my biggest, strongest memory that I might remember. So I started to think about, I don't know, philosophy, religion, but nothing didn't really fit logics. So I decided to find a way to live longer. Back in the time at around 10, I met my current business partner for Cortex, and I told him I wanted to live forever, and he replied that he would rather replace humankind by robots. So 15 years after, I got my PhD in the field of aging genetics. And in the meantime, I really changed my plans, because at the beginning I thought probably I will need to find a way to get frozen in the proper way and then have a second chance later on. But when I started to investigate the mechanisms of aging, I was, for instance, talking to one of my professor in biology, and I asked him what would happen if we tweak a little bit that mechanism in that direction. I think we would live longer. And the guy was thinking a little bit, a few seconds, and told me, yeah, you might be right, but what for? So then I realized that there is a problem that's maybe linked to many parameters that people, they have accepted things, but we are arriving at an age where many things start to be possible. And I completely gave up my cryoconservation thing, and I decided to work really towards like extending lifespan until a stage where we would be able to repair ourselves as fast as we decay. So I think we can start the presentation. So I was looking at data because I had some targets in mind for longevity that I kind of was thinking about during my PhD. I've done some work with this. And the problem is that you might have some targets, but it takes a tremendous amount of time and money to develop a drug at a very low success rate. So I started to do a few businesses in the meantime, thinking about how fast we can do something actionable. So before I was completely against everything that was natural or whatever, but when I realized that some natural compounds are extending the expression, increasing the expression of some genes, I started to be involved in epigenetic nutraceuticals or even cosmeceuticals. But that was just like on the side because I was waiting that the technologies would get cheaper so I could start achieving my goals by starting my research. But then still things were very slow. So I decided to investigate cheminformatics. And then I realized there was a big gap in machine learning. So I contacted my old friend who was back in the time a professor in quantum information. And I convinced him to retire. And we started Cortex. Now we are a team of 5, mostly machine learning, a little bit of quantum chemistry, and we're working on 2 technologies. The first one, it's like an advanced version of what people call QSAR, like quantitative activity, quantitative structure activity relationship. It's basically learning from experiment. The input is a molecule, the output is a property. And we're working on a second technology that is called docking, where we look really at the 3D interaction between compounds. The first technology is already available and works. So I'm going to talk about that one first. So we gathered all the data that we could get, mostly from PubChem and Campbell. It's the largest repositories of high throughput screening data, and also from publication looking at pharmacological data about compounds. So we have about 260 million data points on this database, and we built some neural networks where the input is compound and the output is a property. These are deep graph neural nets, and we train these neural nets to predict every property that we have in our database at the same time. It's about 2,000 properties, but some properties can be anything like, for instance, the number of cells that die, an activation of a target, an inhibition of a pathway. We just optimize actually the compound for a number. So what we are doing in terms of machine learning is like it's basic, it's standard. So we're using, let's say, 80% of a data set where the input is a compound and the output is a property. And the neural net is making a correlation between the molecule structure and the property. And then the withheld data, we just plug in the 20% of the compounds. And we make a comparison between the predictions that we do with these 20% of compounds and the actual reality. And that's what you can see in the red and blue graph. So this is the testing set. So the active compounds are the red compounds and the inactive compounds are the blue compounds. So here is good because the active compounds, we give them a high probability of activity, and the inactive compound a low probability of activity. And the way we actually quantify the accuracy of our predictions is the false positive by the true positive rate, actually the area under that curve. And it's quite interesting because it enables to measure, to compare experiment with different units. It's just about how much you're right or wrong about your predictions. So I'm not going to enter into that table into details, but we've looked into competition. One of them, Cyclica, for instance, that was acquired for 40 million a few months ago. And their prediction ability in terms of AUC for cytochrome. It's this enzyme that metabolizes drug. Their AUC is about 67 to 17%, where we are in between 93 to 95%. A random guess is 50%. So then we've compared ourselves to online tools, actually publications that were making better results than the online tools, and we consistently have done better. And actually, we realized that we start to be as good as the experiment that we learned from. So one of the first application is virtual high throughput screening. So most pharma companies, they will start a campaign to look for a compound for specific a target. They will start by screening, let's say, 500,000 compounds, and then they will select a subset of active compounds. So here this is a virtual experiment where we learned on a data set that was looking into compounds like killing parasite Georgia lambia, and this is our testing set. So actually, if we looked at the top 10% of our predictions, they still contain 99% of the active compounds. So that means that people could test much less compounds if we pre-screen through our systems. We have the potential to squeeze really the size of a real high throughput screening to much less compounds. So there is a reduction in cost and in time. There I have this a fancy example about cytochrome, this enzyme like processing drugs inhibition, and we had the active compound of Viagra in our testing set, sildenafil. We correctly predicted that it was being processed by that enzyme, but that was not luck because our AUC is 95%. So then we have been also including like properties such as solubility, log-P, permeability. We actually are able to predict most of the pharmacological properties that are being tested in the preclinical trials. So here is again a virtual example about an experiment that we would do to find compounds against malaria. So we had a chloroquine, which is a drug against malaria that was in our testing set. We correctly predicted that it would be active, but since our system is predicting every property at the same time, we could see that at the same time it doesn't affect DNA repair, the nervous system, or liver function. So it could be a good drug candidate. Then we also have been kind of rediscovering drugs in the field of parasitology, bacteriology, oncology, and virology. So in our testing set, we correctly predicted the compounds that are being used as drugs or that are in trials as drugs. So the technology is therapeutic field agnostic. You might think, okay, this is very nice, but this is all virtual. Maybe Ivan is lying. Maybe there is a leak between the training and the testing set. So we had to do an experiment. It was during COVID, and since we predict many things at the same time, instead of looking at only one type of experiments that was consisting of looking at compounds that are inhibiting COVID in SARS-CoV-2 infected cells, I decided to look into also assays that were reporting measurements of compounds being tested for inhibiting host cell protein that are being hacked by the virus or directly viral proteins. We can also predict toxicity, fluorescence, so that enabled us actually to successfully achieve our first proof of concept in the laboratory. So I convinced Professor Olalier in Aarhus University to test only six compounds that were actually our best bet regarding all the compounds that were in clinical trials or being used as drugs. They were never tested for COVID, and 5 out of the 6 compounds were antiviral, and none of them were toxic at the active dose. And now we have 2 of these compounds that are further in the preclinical phase. They work with different variants, with different types of cells, with lung cells, with patient cells. So we are in the process of patenting and publishing. We've done some other proof of concept that was exactly the same training, but we gave a few other compounds to Stanford University. They had another readout that was fluorescence, but we were measuring the same thing, and we also discovered new antivirals. We have done that other very different experiment. It was about compound selectively killing cancer cells. So here, with the Victorian Center for Functional Genomics, we have had a little test. So they gave us 80% of a data set of about 5,000 compounds that were either active or inactive in terms of killing or not killing cancer cells. And our predictions were tested on the 20% remaining compounds. And actually, we were as precise as the experiment reproducibility. That's actually led me to start some collaborations. I mean, it's ongoing, but nothing is produced yet. But I'm talking to a few of the main guys in Senolytics, because if you are able to predict out of a small data set compound selectively killing cancer cells, why not senescent cells? So this is something that's ongoing. And then we've just done something that's more actually in order to show that we can offer this as a service, because we've been doing some things on compounds that we chose, but actually we wanted to show that we can really be useful in terms of large screening. So we commissioned Syntef, is a Norwegian company, and the Norwegian Center for Molecular Medicine to test a library of compounds. So our system, actually, we looked into a library that was available for them, where they should be the largest amount of GP41 HIV1 inhibitors. So GP41 is a protein of HIV1. And actually, we believe there would be about 22 hits. We committed to sending our predictions before they do the test, so they can support us in terms of like how good we did. And actually, we had 20 out of the 22 hits in our top 10%, and 2 hits in our top 3 predictions. So the probability we get there by luck is about 1 out of a billion. So the technology works. Then I've been talking. I'm starting to talking to pharma companies, and chemists were worried about how new the molecules can be. So we started to make measurements of distances between compounds, and we realized that the closest active compounds in our training sets compared to the predicted active is actually further than the closest inactive. So that shows that we can really find compounds with new properties. The neural net is not simply stupidly copying some motifs. We are good at generalizing and finding new families of molecules. So in terms of practicality, high throughput screening is implemented in the pharma industry. You also have high throughput screening centers. So people typically will test 500,000 compounds. It will take them like anything between 3 weeks and 6 months. The cost is about $1 million. And what we can do is to propose them to actually screen on 10 times less compounds, learn from that data, and then virtually screen not only 500,000 compounds, but all the compounds that you are actually able to purchase. So 23 million is actually the reliable providers that we contacted, that we knew were reliable and who were fast enough to send us the data. So we can implement any new library. Also, we have this collaboration with one of the main molecule providers, and they have a data set of a virtual library of molecules that they can actually build. And we've been creating a system, de novo compound generating system, that enables us to put the fragments that they have together and to reach it's in the range of 50 billion compounds. So we are sure that for customer or for research, the compounds that we want, that we virtually screen, they can be then physically produced. So that's pretty important. Then just an idea about the scope of this, like the accuracy of our prediction is really what we focused on. And as you can see, the prediction accuracy is increasing the number of hits. So we can clearly show the value of our service, because the hit is in between 50K to 200K. But with the accuracy we have, that's on par with experimental accuracy, we have about 5 times more hits than what you would do traditionally. And these numbers are based on the statistics that were actually correct with our proof of concept that I showed before. So we are launching now a service that's a little bit easier, because the virtual high throughput screening will actually force us to get data from the customer of our customer, because a high throughput screening center is doing it for a third party. And even though we don't need to know the target activity, it has been a little bit complex, because it's a three-party contract. So another thing that we can do, and that we're happy to do for any biotech, especially working in the longevity field, is that they can just send us a file with the compound structure. So there is a very simple way of doing it. The name of the way to encode the compound is called a SMILE, and out of this we are able to predict most of the pharmacological properties. So if they have a list of compounds, they can already pre-select the compounds that will not be toxic and that will have the good pharmacological properties. So that's very interesting, because that's just a part of the service. But what we can do, since we can screen 10,000 compounds per second per GPU, that means a billion per day, we can not only screen the molecule for its ability to activate a target, but we are also able to look into off-target effects and to select compounds that will have that will be in the range of the good pharmacological properties. For instance, if you want some drug to access the brain, you want a good permeation of the blood-brain barrier, and so on. So at the moment, we are working on 2 things in terms of research and development. So we are curating our database for the first technology, and I started to initiate partnerships with key figures in the longevity field that have access to laboratories, or that have laboratories. And so we have targets that we have curated, and we're about to send our first longevity compounds to be tested in vitro and then in vivo in C. elegans. So we've been looking at targets such as mTOR, NRF2, HDAC, HISP, heat-shock protein, and so on. And these are not my favorite targets, but they were in our database, and we have good predictions for them. So this is something that we are currently doing. We have also assays for autophagy that we can predict very well. And as I mentioned before, we have some project with Senolytics, and I'm trying to also initiate some project with a few people regarding high throughput screening regarding DNA repair, because my big problem with our database is most of the DNA repair targets. People are looking for compounds that actually screw DNA repair because they want to kill cancer cells. So we will probably be able to find some compounds with a negative result, but that's a little bit tricky. So I'd like to have a project where we can generate lots of data, let's say a high throughput data level for to find new compounds that will be enabled that will enable to trigger DNA repair. Also, we are curating everything that's linked to age-related disorders. So usually the way we collaborate, we've done that with Stanford, and we basically tell them, here are our compounds that we can send you for free. You test them for free, then we share the IP. And out of this, if something happens, we can start writing publications, patents, and then look for money from grants or basically create a spin-off. And most of the people we've done the proof of concept with are doing such things with us at the moment. Then there is once we get some more funding, we would be able to work on transcriptomic data. That would be very interesting, and also on high content imaging. But that's another story. This is really research and development. There is another research and development thing that we're doing, which is actually advancing very fast, and I'm excited about it because not long ago we just had a prototype that we call a conformer that folds small molecules faster and more accurately than the state of the art. So our approach is to learn from simulations and then to kind of use that data to feed our deep neural nets in order to do good predictions. And actually, here we optimize for the energy above ground state. And you can see that we are better than the state of the art. So we predict lower energy for the conformation. And now we have a docking prototype. We're about 1,000 times faster than current tools. And our preliminary results show that we are as bad as them, actually. But very soon we should be able to have a working tool, because that was only a prototype, and we have several things we can optimize on. We basically implement some force fields that are approximations of like Schrödinger's equation, and we are choosing better force fields, and we've done that with kind of shallow nets compared to our deep neural net. So very soon, we're very confident we will have a docking system with an unprecedented accuracy. So now that's probably more of a pitch slide, but people would tell me, okay, what's the difference between what you do and the others are doing? Is that I think I want to insist on the fact that we are the first to be as accurate as the experimental reproducibility. And that's very important, because like the high throughput screening market is 18 billion, but then like this is not only high throughput screening, because we can predict for pharmacological properties, we can parallelize things and really prevent a lot of back and forth of compounds that are toxic. So then they have to create new compounds. We can generate analogs of the hits and then test them virtually before they have to be produced. Meanwhile, the competition is surprisingly looking into, for instance, generative AI. And here it looks like you want to generate, let's say, look at a ligand, a drug as a key, and the target as a lock, and they want to generate new keys, but actually they have no good way of predicting how the key will interact with the lock. So without this drug target activity guidance, we don't think that is really efficient. Also, there is lots of language models. I mean, they are powerful, like in other areas, like ChatGPT we have seen, but with language models, you're unable to see the difference between your left and your right hand, and so you can do nothing about chirality. Then regarding docking, people are still looking into crystallographic data, but crystallographic data is very scarce. You have maybe 40 compounds for one target, and then also it's not so accurate because the compound is not stable that way. It's like moving constantly. So and finally, 40 compounds per target doesn't help you. With deep learning, you can do nothing. So here, by generating data with the force fields, and then kind of squeeze, like learn from that and make this computationally faster, we are able to harness the power of deep learning to already make good predictions. So in terms of business model, I mean, I don't know if this is really in the scope of like this this talk, but so we are basically trying to work with people in the longevity field and co-develop a pipeline of longevity compounds. So far we are starting to send our compounds to be tested in different areas. So I'm choosing people who work in different areas of longevity. So like it would be like senolytics, DNA repair, any specific expert of some targets. Then also we need to feed the company. So we're starting a service where we can virtually test compounds for their pharmacological properties. We can also do the trick with the virtual high throughput screening by learning on much less data and allowing people also to test 10 times less and get actually more hits. And I also initiated some conversations with the pharma industry, where I would like to have more of a milestone-based project with a small upfront payment. But since we know the technology works, we would like to have like some kind of success fee when the compounds are reaching a specific phases. So we will focus mostly on the preclinical, of course, because after it's another story with the clinical trials. But still, I think the time for the whole preclinical phase is about 2 to 5 years. Here we could squeeze that to 2 to 5 months actually. And regarding the what we have done for COVID, usually it would take a few years for a few groups together to work on that. Back in the time, we were 2 guys and we worked on it for a few weeks. So so the cost is quite negligible. Then at the moment we're looking for 7 million for 2 years just to cover our costs. I think we should be profitable next year already, and so we planned on being like among the main players with the better technology, but still less resources. So of course we need to spread the word and put a foot in the pharma industry, which is quite difficult, and lots of the budget will be linked to actually having business development guys who have a foot in the pharma industry to just open a door to give us a chance to show that our tech works. So like the take-home message regarding the technology, so regarding what works and we can already offer as a service, or that we're doing through collaboration, is that we have a technology that can literally save hundreds of millions and years in the development for each drug. It is proven, it has been tested in different laboratories. It is fast and accurate. We can test 10,000 molecules per second. It makes 1 billion per GPU per day. We learn at every property at the same time. So we are able to parallelize heat discovery with lead optimization. So not only we will look at a compound that's potent, but we would be able to select by the probability of being toxic, not having some specific off-target effect, and so on. And then the technology is in any, I mean, it is therapeutic field agnostic. But the idea, of course, since my main objective is to survive, all what we do in-house is to develop our longevity pipeline. So I think that's it for the short presentation. So I guess you guys will start asking me questions. I'm sorry, there was something in the chat there. I'm not sure if something was for me, but I didn't read anything. So
yes, so there are some interesting questions from our viewers as well as our panelists. So I will pose this one from Prosper, who wonders, can perhaps some of the compounds that you are testing virtually lead to a cure for HIV? As we know thus far, essentially the options that are available for people are treatments. They can lengthen the life expectancies of people who are afflicted with HIV, but not quite cure the disease. Do you think there is a compound out there that just needs to be subjected to testing, needs to be identified, that could actually provide a cure?
I'm not sure there is a compound that already exists, but definitely with our system, we should be able to generate a compound that would do the job. Because now with our small test with the GP41, the HIV-1 protein, it's a protein that helps the HIV-1 to enter cells. We have been able to find a potent novel antivirals. So I believe that it would definitely help, and it would be possible. Of course, we never know because this is R&D, but my first guess would say yes.
Yes, thank you. And along these lines, in terms of the process involved in using this approach to synthesize new compounds as compared to testing the effects of existing compounds, how many more steps would be involved and how much more time would it take if you were looking to synthesize a novel compound and use this approach as part of the initial identification of the structure of that compound, depending on what you wanted to do?
2 weeks with the provider that we are talking to.
Nice, nice. So there's not that much of a difference between evaluating what already exists and creating something
No.
completely new.
Yeah.
And it's also interesting to me that you mentioned there are some natural compounds that you've been interested in studying. Do you have any insight as to which ones are actually effective or promising? And is it the majority of the natural compounds that are sometimes advertised, or just a few, just a small subset that actually have beneficial effects?
Okay, so we've not done our first screening regarding our longevity pipeline for natural compounds. It is in the process, so within a couple of weeks we should have that. But I've made an effort because I want something to be quickly on the market. So we have all these libraries, and we have kind of classified them by natural compound, FDA approved, natural compounds, drugs that are like in clinical trials or that are approved, and any compound that you can purchase physically from a library, and finally any compounds that you can synthesize. So all of these will lead at like very short term, medium term, and not too long term, because we really hope to accelerate the whole process. And so yes, and there will be some compounds during our HIV thing. We've seen some fancy compounds appearing there. So no, there will be some natural compounds. I believe they are not going to save our lives, but they might be enabling us to reach that escape velocity. I mean, I don't talk about this way, but it's actually I have the same vision about it. So the faster we can eat something that can makes us live significantly longer, the better it is. So I will definitely look into directly trying to work with the food supplement company to directly implement the compounds in something that's commercializable. They should be better than what we have today. Yes.
Yes, indeed. And anything that gives us a few additional years would also increase our probability of reaching longevity escape velocity. So
Yeah.
it is all in the interests of moving in the right direction. Now, Alan Crowley has an interesting business-related question. He notes that your company is organized as a GmbH, which in German-speaking countries is essentially quite similar to a limited liability company. And he wonders, how does that influence national and international investments?
Not that much actually. To be honest, I did set up that company during COVID. We didn't know where to go. My business partner was homeless. He was in between South Korea and Germany and Hungary. I was in Hungary. We needed to find a way to set up a company where we would have, let's say, the potential to attract some talents, and where something is happening in terms of in terms of biotech and pharma. So I think that as a German company in Munich, it's quite a good image, and let's say investors that invest internationally don't have problems for that. However, I know there are some specific grants that are only for American companies. I mean, initially I wanted to go to do that in the States, but during COVID it was impossible. So yeah, to make the answer short, it's not really a problem as a German company. Specific investors would only invest in specific areas, but most investors in that area are probably really, let's say, area agnostic.
Yes, thank you very much for that answer. And we have a question from Daniel Tweed, who wonders, to what extent do you let pathways to marketability drive your research decisions versus doing promising basic research?
It's a compromise. So I was not so interested in the COVID, but it was easy to do the. It's just some people at the beginning was thinking, oh yeah, but this technology is just for repurposing. No, it was for repurposing because it was cheaper to purchase the compounds that already exist than to synthesize new ones, and we had of course less chances of them to be toxic because they are already on the market for other purposes. So we will always have to do a compromise, because if you want something that actually lives, it needs to exist on the market. So we are really striving to do some fundamental research, but of course we need to live to survive, also to make some noise. So it is a compromise.
Yes, fair enough. And I think every research-oriented business makes that compromise to one extent or another. Daniel has another question. He wonders, when was quantitative structure activity relationship modeling first developed, and approximately how large is that community?
I'm not a chemist, and I know that has been there for a long time, but the technologies that are used in terms of like understanding the data, it was not even machine learning, or really obsolete machine learning, to like shallow neural nets or like random forests, super vector machines, and so it kind of gave a bad reputation to QSOR, and people, they did not give up, but in the pharma industry and also in other research areas, they just don't emphasize so much on it, and they use it as a, not as a guide, but just like something that might give them some insight. Meanwhile, if you reach experimental accuracy, this is like the main tool that people should use. So, but that was really enabled by our hard focus on being very precise at this. So we had to let's say get inspired by vision recognition and to use the latest machine learning models. So actually, QSOR itself is like like predicting an activity out of a molecule structure, but the way you predict it is you have a million ways of doing it, not a million, but many ways of doing it, and I think we found the strategy to make QSOR useful. But I have no idea. It must be something like 20 years. But it was basically useless back in the time because the statistical method were not good enough and there was not enough data. Now there is more data and it will start to be. I mean, now through us, it should. I believe it should be the first thing that you should do in terms of screening.
Very interesting how the availability of data makes techniques that were previously developed but perhaps not had as much application quite a bit more useful now. So thank you for that answer. And Daniel has one more question. What computer languages do you mostly use, and is Python suitable for this kind of work?
Yeah, Python.
So this is actually a language that many people know and have at least some basic experience with, and it can be harnessed for such quite remarkably advanced applications. So thank you for that answer. Now, I was also curious to get your thoughts on Google's, Alphabet's AlphaFold. DeepMind is a subsidiary of Alphabet that released this artificial intelligence system called AlphaFold for the identification of protein folding configurations in a much more effective way than the previous approaches, which were essentially using computational brute force and distributed computing across millions of individual users' computers. And AlphaFold, my understanding is it's not perfect, but it can accurately predict a lot of protein folding configurations. So when you compare and contrast your approach with AlphaFold, what can you say about that?
So I like the concept of AlphaFold a lot, actually, because we will be able to use their technology in order to look at when our docking system will be finalized. We will use their technology in order to have the 3D structure of our target. So that's extremely useful. But what they do actually is machine learning on data that comes from sequences evolution. So it's very different, and there is not much about how things are interacting. So it's a different technology, but it's a technology that's extremely useful for research and for us in our docking once it will be developed in a few months. Mm-hmm. The main message here is like really AlphaFold. They're really good at making these protein conformation, but they don't look into interaction between things.
Interesting, but I'm glad to hear that there is some complementarity between those technologies. You also mentioned how your technology compares favorably to generative AI and large language models. And one problem that even layperson users have observed with those approaches is sometimes they can just make things up through what's called hallucination, where they essentially create facts that are not actual facts, but they seem like they might be plausible. And to someone who's not an expert in the field, it's very difficult to distinguish those hallucinations from actual facts. So that's a vulnerability that I see of using generative AI for drug discovery, because it could just invent some sort of fictional compound that looks very good on paper, except it doesn't exist and cannot exist. So how would your approach prevent that kind of outcome?
Okay, so actually regarding our approach, we are creating compounds, but at the moment we really want to make sure that we can synthesize them. So we virtually screen compounds. There is some kind of evolutionary system that will enable us to screen even more than 1 billion compound per day. But actually we build the compounds in a way, in the same way that they are built by the companies that can produce them. So actually we can come with like a very large number of compounds, but we make sure that people can synthesize them, because otherwise it will take too much time.
Yes, and that makes sense. What kinds of criteria do you consider when you think about whether a compound could be synthesized?
We don't really. I mean, there are two things. I mean, there are systems that show basically if if the thing would explode or not, and on the other hand, like we just tap into libraries of compounds that either exist or that the chemists who are actually offering the service of synthesizing, like in this virtual database of lots of compounds, we make sure that we synthesize in that chemical space.
Yes, thank you. And more generally, when your system predicts certain effects for a compound, what kinds of empirical tests are necessary to ultimately determine the validity of that prediction? So, for a lot of the properties, you said your system has accuracy rates in the 80s or 90s of percentages. What actual tests in a lab would ultimately confirm or disconfirm the prediction?
So it's rather in between 90 and 95, and yet it's the AUC. And then actually we're getting better when we send compounds, because as we have done with COVID, we are actually selecting upon different predictions. So we have built that system that actually allows us to make an algorithm of all the predictions. So we don't only look at experiments that look at is this compound active for this, but also other experiments that can lead us to think that the compound would be active as well. And it's pretty simple. It depends on the type of experiment. So, but it's just a question of sending the compounds into assays or into cells and see if they're active or not. So you have like let's say how can I explain this? Well, it's basically standard assays. Like it's a depending on the experiment, it's going to be €1,000 to €10,000. So it's nothing crazy. You just test and retest the compounds, and you make sure that you have some specific compound here that is like usually active, and some specific compounds that are usually inactive. So you just make sure that there is no problem with the experiment. But it's a standard assay measurement, and it's not expensive, but it really depends on the type of assays that you're measuring. With for GP41, we've done that on a larger scale because it was a molecular assay and it was optimized for like a small high throughput screening. But for COVID, it was just cells that were like infected by SARS-CoV-2, and the compounds are just thrown them, and then you look at the amount of RNA. But it's pretty straightforward and simple.
Yes, thank you for that answer. And I'm also curious in terms of what have you observed the common sources of error to be? In other words, how can reality differ from the prediction, and why do these differences exist even though you're trying to minimize them, and how can they be minimized further?
So far, I mean, we're kind of happy to be as good as the experimental accuracy, and then we did not see any patterns in the compounds that were, let's say, in the few compounds that were not considered as active. And I think that was falsely considered active. And unfortunately, that will uh, most of the time, I guess, be experimentation related. So if our experiment is very accurate, we will be very accurate, and we might even reproduce the same type of error as the experiment. But we have not seen that. So I'm just like thinking in real time. But so far, we don't have any direction into specific types of errors. We're just happy to see that our results are reaching experimental reproducibility if we have enough data, and if, of course, the training data is not an analog of the same compounds. We need compounds that are a bit different from each other, so the neural net can build a law between the compound structure and the activity.
Yes, thank you. And you spoke about also evaluating the off-target effects of a compound. Could this be used to, for instance, discover hidden side effects of certain drugs? Even drugs, for instance, that have been on the market for a long time, but sometimes we've seen the FDA recall certain drugs because side effects were discovered several years after they were approved, and those side effects ended up being a lot more severe than previously thought. So could this approach perhaps shed light as to perhaps hidden adverse effects of certain drugs that were not considered before?
Yes. The objective is to do that in advance, of course, because you don't want people to have the side effects. But definitely, this is something that we could do. Yeah, we are actually creating our whole database, and then once we have all the targets that are usually being screened, we will be able to predict any standard of target effect. We have already some of them in our AdMed service, because you don't want like this. There is a specific receptor, ERG, that's going to create like some heart disorder reaction, and that's something that we can basically predict and prevent.
Yes, and I think that would be of great benefit to a lot of people. Our friend Didier Cornell has a provocative question. First, he says, your presentation is interesting and clear. He wonders, why not work in collaboration with Insilico Medicine, which is also using artificial intelligence for drug discovery, rather than be a competitor? He thinks it seems you have the same goals and motivations. Or I wonder if you have a different perspective. Perhaps your areas of focus are different in some way, or your systems are different in some way. But what do you think of this question?
I have not. I have nothing against Insilico Medicine. I just don't believe they have the right approach, and they're spending a lots of money. I mean, this is not. It is not impossible. I mean, I wouldn't completely refuse a collaboration with people in the area, since the objective, the ultimate objective, is to accelerate drug discovery for longevity. But so far, I mean, it might be. I mean, I'm not saying no. I'm saying it's a good idea. It might be dangerous from a business perspective, but yeah, it's not. Yeah.
All right. Well, thank you for.
I know Didier Cornell, actually.
Yes.
Yeah.
He has been a frequent guest on our virtual Enlightenment salons as well, and he always asks interesting questions, definitely worth considering. So now I will go to our panelists and see who has additional questions for Yvonne. Let's go to Ben Ballweg.
Wanted to follow up about AlphaFold. I think I was speculating that you wouldn't be able to practice or to even have Cortex without AlphaFold having come along, but your answer made it seem like, you'd have been just fine if AlphaFold hadn't existed. Could you clarify how useful AlphaFold is for you, or if it's just like, our system doesn't use that at all?
Okay, so the first technology does not use AlphaFold at all. Like we train from data, like the input is a molecule, the output is a property, and we're good with this. But for targets for which there is no data available, you need to either create the data through high throughput screening or proceed to docking. But docking doesn't work so well today. So for proteins where you know the structure, we could actually use crystallographic data, but there is no crystallographic data for all proteins. So actually, AlphaFold will be useful for us to expand the docking to any target that actually is interesting for us. So it would just like give us a big hand for the extension to the docking to let's say poor known targets.
Yes, thank you for that response. And now let's see if Art Ramon has a question.
Yes, in this creation of molecules, do you ever run into a molecule that has a really negative effect? Like it just rips apart cells or anything like that? That's novel. Anything that comes out from your research there?
We have not really. We were rather digging into selecting molecules that are not very toxic, but definitely regarding. I mean, no, we've not encountered this, but this is something that could be found. Yeah. I mean, the idea is that if you want to selectively kill cancer cells, you don't want something too aggressive to kill the all the cells that are surrounding the cancer. So yeah.
Yes, thank you, Daniel.
Yeah, really great information. How compatible do you feel your work might be with upcoming advances in nanotechnology? Molecular docking sounds an awful lot like molecular level machining. And is that something that you feel you're positioned to take advantage of if and when that becomes more feasible?
Yes, this is something I clearly have in mind from the beginning, that we started to work on docking, because these molecular machines, we are molecular machines, right? So because people have this idea of like small robots that are metallic or whatever, but I believe that the most efficient nanorobots will be kind of built the same way that we are built. So actually, like predicting how good some specific shape is going to intercalate into two specific bases or like amino acids, or enter into some specific receptor is going to be useful. So definitely, I plan on being involved in such project when the opportunity is here, when the docking system is working.
Do you think that kind of nanomachine repair is the holy grail of longevity, perhaps? Or any opinions on when we'll get that date-wise?
Well, so far, I would look for compounds that help to increase the repair that already exists. Then new repair machines. I mean, it's not science fiction, but it's not in my plan for the next, let's say, 5 years. But it's not science fiction.
Yes,
I just
that
don't
is
see how to do it right now.
Very good.
Yes, that is quite intriguing. And of course, a stepwise approach is reasonable, but I'm glad that you do see that future of essentially nanoscale repair as being attainable, because that could undo a lot of the damage at the cellular level in a way that can just recur periodically. So people wouldn't have to have like massive surgeries or other kinds of, let's say, potentially traumatic interventions, they could just have the nanobots, biological nanobots, repair their tissues on an ongoing basis. So that is quite intriguing, but perhaps not in the next decade or so. Didier?
Yeah, I wouldn't say I said 5 years, but like because things are happening, but we don't know. But so far, I guess that if we find better senolytics and we can like harness some kind of like healthy young cells, like destroying the old, bringing some young, and then like also giving medication to the body to slow down the aging through various like strategies such as like reducing oxidative stress, increasing DNA repair, and so on. Is the first step. Then nanotech is the step after. Docking would help, like especially like nanobots that would be made out of like let's say biological, let's say organic molecules. But I think the definition of nanobots, not nanobots, but like I think we will go really close to the molecular machine that we have and just enhance them a little bit. So will it be a nanobot or not? Like that's a question of definition. But imagine that you do a transgene with a DNA repair system that's a little bit enhanced. Is that a nanobot? So just a question of definition, because there we're not too far.
Interesting. So we may have a kind of subtle emergence of nanobots when molecular machines essentially progress to that point where the distinction becomes blurred. And Alan Crowley writes in our chat, yes, Dr. De Weber, science in the world is better than science fiction. So thank you for that comment, Alan. And Didier also says, thank you for the answer. He writes, in general, I think more cooperation, open source activities, and less competition are better for longevity. This is also coming from his left-wing idealist side. I think it's worthy to hear some of your commentary on this. I actually raised this question at the first Longevity Summit Dublin in 2022, where I observed that up until now, the longevity field has been rather collaborative. Most often, there hasn't been, let's say, cutthroat behavior observed in it, quite in contrast to what has happened in some related fields. For instance, in the field of mRNA vaccines, there is now quite a strong patent battle between Moderna and Pfizer, and Moderna sued Pfizer in the middle of the pandemic, essentially, alleging patent infringement. So clearly, there wasn't an attitude of having a shared goal and trying to solve the same problem using a collaborative approach. There was more of an attitude of essentially getting ahead of one's competitor at all costs, even if it risks undermining the progress of the field. So how do you think it might be possible to maintain a more collaborative outlook within the longevity community so that we don't have some people essentially undermining the research of others or making it more difficult to make advances and commercialize advances?
Yeah, undermine the projects of others is like nonsense if you really are in the field of longevity and you really believe that I mean you want some results, because I mean I'm happy to help any company developing some drug if we like with any type of like the prediction that we can predict and so on. But I had this same type of question from Didier, but I also wanted to answer him something just that is not really linked to longevity, but in general, if you look at the landscape of DNA sequencing, back in the time, it took like 50 years for the world and like over 1 billion to get to the sequence. And Craig Venter got about 200 million. and just because like things were well organized in a let's say much smaller but more organized structure, he was able to do much faster for much cheaper. So actually, also if you get the money in the right hands and it's done the right way, it might also be better. So the idea is that at some point, maybe some companies, including us, if we believe that we can be better if we get some part of the stake, then to develop faster to something new, it would be beneficial to keep some of the data not open source. However, I'm very happy to collaborate with different companies and to share data with them, and we are doing that, like training from the data or like sending our compounds to them. And I think in terms of drug discovery, especially because we don't have the vision of the pharma or the chemist people. I mean, I'm a geneticist. My business partner is a quantum physicist, and actually we believe that the scope, that people are happy about their libraries of compounds. There are so many compounds that you can make. It's a little bit crazy to be attached on some compound or compound family, because we can do like almost an infinite number of compounds. So I don't see the competition being that hard in that field, especially that people are fighting towards the same direction. So I would encourage like more collaboration, but I'm just still saying it's not like some data, of course, for the survival of the company cannot be open source.
Yes, and that is fair that a certain balance needs to be determined based on the revenue model of the company in question. Alan responded to Didier and said that he is with Didier to a degree, but we do have to make longevity pay so that people will pursue it. So a similar thought. Alan also recommends a book called Where Good Ideas Come From. He says this book does an excellent job of showing where collaborative versus individual and secret versus open research produced the best results. And here is a link to this book. It can be found on Amazon. The author is Stephen Johnson. So I would encourage anybody who is interested in evaluating the relative merits of these approaches to check out this book. Alan, would you like to elaborate or perhaps ask a further question of Ivan?
Well, sure. I'd like to elaborate on the book Where Good Ideas Come From, because I thoroughly enjoyed it and I've recommended it multiple times in this forum. And it basically has 6 chapters about influences and aspects of existence that bring about good ideas. I mean, it uses a continuous metaphor throughout all those 6 chapters and the intro and finale that it has, which is goodness, the guy that invented evolution. His name escapes me right now, but because Johnson did a study of his journals prior to developing the theory of evolution, and uses that analogy along the way to show how sometimes it takes several different factors, what he calls the slow hunch, and not the aha moment. So, this building upon of ideas over time, we like to characterize it as the aha moment when we say, oh, here's what we need, nanobots. Whereas actually we started with giant industrial machines, and we've slowly progressed toward nanobots. Anyway, the entire book is fantastic, and the last concluding chapter does show a relationship over time and a matrix that involves those variables about when and where those things work best for innovation and development of good ideas. Now, that's the book. But going on, I've got a question for you. I think your work sounds fantastic, Dr. Weber. This idea that you're researching the proteins, it sounds to me almost like that is the first step before finding out combined results, the pharmacological results. So what do you have what's the pipeline? Do you provide the seed of an idea? Here's a protein that can be developed and studied, and turn that over to another organization, or do you care? Does your organization follow that all the way downstream, so to speak? And I'll stop.
Okay, so unfortunately, no. First, we are only at the moment working with small molecules, which is most of the drugs. So small molecules that will activate targets that could be proteins or that could be a pathway, because sometimes we can do prediction without even knowing the target. So to let's say target an effect. And actually, that's the idea. I mean, there is something that we will, let's say, not have open source like our code for our AI, because I mean, we will not be able to eat without it. But on the other hand, I know that I won't be able to be bigger than 10 times Pfizer within the next decade in order to produce drugs to make myself live longer. So I gave up that idea since years. So the idea is really to have the compounds that have a very high probability to work, and to send them to the right partner who's able to validate that these compounds work, and then with these partners look for other, like let's say mostly financial partners, to push that further in order to then get some other funding to go through the clinical. So really, my objective within the next, let's say, 4 or 5 years is to have, let's say, 200 compounds in 50 companies that are actually progressing, like at the end of the preclinical, or like already advancing in the clinical trials, which are with a much higher probability of success. So that will not be done by one entity. So I completely agree. Here, especially for longevity, we need the collaborative approach. That's it's just necessary. So and then that's our strategy.
Thank you.
Yes, indeed. And that truly does speak to the value of a collaborative approach, because when the entire longevity field makes advances and prospers, we all would benefit. Didier writes in the chat, he's sure that private organizations can be better than public organizations. However, if you do not share the results, it is not useful. In theory, patents are for everybody, and only profit is private. Practically, however, he writes, patents are often made to not share knowledge. And he says to Alan that actually longevity would pay by giving longevity to people, and extraordinary fame for those who find something useful. Well, I can say to Didier, my own motive for being in this field is clearly not monetary. It is to not die, because I see life as the greatest wealth. And of course, the longer I'm alive, the more opportunities I have to do anything whatsoever with my time, including making money. Now, I am not in a position where I am running a business myself, though, but sometimes, as you pointed out, Yvonne, businesses also benefit from collaborative approaches, and to the extent that patents exist, I agree with Didier, they should be encouraging innovation rather than discouraging innovation. So a question arises from this. Have you seen a situation in the longevity field or in a related field where somebody was holding on to a patent and preventing others from utilizing it, preventing others, even if they wanted to, say, pay a small or reasonable royalty, from actually using the technology or the method that that patent covered to make further progress?
I've not seen that myself. There was just this guy at some point who kind of like stole some institutes and companies' money because he bought some patent on BRCA1 and 2 genes for sequencing. But that thing was extremely blurry. It didn't work too long, and I think it was a scandal. because this is just for diagnosing. I mean, also like it's really predictive in terms of breast cancer tests. So no, this is not something that I've seen besides that specific example. So.
Yes,
But
thank you.
definitely it is going to become more pharma, because before, like, longevity was snake oil. Now it's getting real, and so we will be like fighting the same fight as pharma. So there will be some struggles. But the idea is that at some point, if you have some idea, some vision, some plan, you need some cash in order to push that plan to the reality, and to make that cash, you need to make some money. And if you don't have any IP, you won't be able to get that money. So, but of course, like my strategy is mostly collaborative.
Yes,
and
indeed.
I would be happy to have a competitor finding longevity compounds thanks to a collaboration with us, even if we're just a service provider and get almost nothing out of it, because I'm going to eat the compound. So like that's the big difference. I'm just saying I need to keep a little bit of the IP in order to grow, but then, like, of course, the whole work, the whole development is collaborative.
Yes, indeed. And that's understandable. Alan writes that, yes, the benefits of health span and lifespan are valuable rewards, but the research is expensive. And Alan says he would gladly pay for better memory. Mike Lazine writes that automakers and others have done this kind of thing for years. They snatched up patents and inventions and then sat on them. So this is more of a problem in more established industries or more conventional industries. Big Pharma has a bit of that problem as well, but I'm glad, Yvonne, that you haven't seen that as much in the longevity field, which speaks to some of the unique dynamics and incentives in that field. I just hope we can keep it more collaborative and we don't get a situation where all of this research essentially falls within the power of some conventional pharma executive who then says, oh, we can just sit on these patents instead of allowing this set of discoveries to come to any sort of practical application. There are some who say that Big Pharma is incentivized to maintain the status quo and sell people essentially maintenance medications for their conditions, but they are less interested in pursuing cures, or if they have a blockbuster drug that kind of maintains people in a tolerable state of health but doesn't cure the underlying disease, and there's this new compound that could cure the disease for good. Some people say, well, the pharmaceutical company's incentives would be to get the patent for that new compound and just sit on it, not manufacture it. But what do you think about that? Do you agree or disagree that that would be a powerful obstacle? Or do you think there's enough of a profit opportunity that big pharma companies would more likely jump on true longevity medicines or true disease cures and prefer them over the maintenance types of medications?
I think if they have something that works better and that they can finance it, they would go for the better product. But definitely they have to sell what they produced. So like that's why sometimes you are a little bit surprised about the little amount of side effects that were kind of discovered during the trials and then what's happening in reality. But definitely, I believe that if they had such a compound, they would like try to push it further, because there is basically more money in a compound that will work better, and it's also marketing and reputation. So I don't think they would hold onto compound too much. And actually, as I'm saying, compounds for me actually they are not very important. I think there are so many compounds. What counts is the technology that finds the type of compounds that work. But one compound is not magic. If there is one magic compound, you can find another 1,000 magic compounds. So these patents are just basically protecting the guys who worked to produce them, so they can have a little bit of cash and continue doing their research. That's the way I see it, or that's the way we would do it.
Yes, thank you for that perspective. And our friend John H. writes that within his own lifespan, science and science fiction have gone from the realm of the nerd to the general public, and this is quite amazing. The downside is that he, as an old sci-fi nerd, feels so much less special. And I was curious, Yvonne, to get your perspective on this because, like me, you were interested in defeating death from a very young age. And compared to your expectations of the progress that science would have made by now, how has the reality matched the expectations or diverged from the expectations? Are we further along now than you expected when you were a child, or are we perhaps behind? Have certain advances not happened as quickly as you would have hoped?
I wouldn't think about myself as a child because I really didn't have much data about, I mean, to make myself a critical opinion. But as a young adult, I've seen things moving faster than I expected. So I'm more confident that it will happen than I was before. I remember when I started the university, I thought we'll find a way to get frozen and have a second chance. And then during the course of the university, I decided that there would be a way to kind of avoid this like cold sleep by just like prolonging lifespan until we find a way to repair as fast as we decay. And that is not too far, I believe. So if we can have the same type of conversation in the same type of physical shape in about 25 years, I think we're done.
Yes, indeed. Well, I would agree. If in 25 years we look the way we do now, clearly something will have happened to enable that, some sort of progress in medical science. And based on the logic of the concept of longevity escape velocity, that is pretty much what we would need to do, and of course avoid accidents and self-destructive behaviors in order to not have an upper bound to our lifespans, in order to potentially live indefinitely. So I'm glad that you are more hopeful now than you used to be. You said previously you had been focused more on the prospect of cryopreservation, but now you seem to think that people of your age would be able to avoid being cryopreserved altogether, at least in terms of the probabilities favoring the outcome of just rejuvenating oneself. Would that be correct to say?
Yes, I mean, I'm going to fight for that. I'm going to fight not to be in the cryo tank. That's the idea. But another thing regarding this is a little bit of a philosophical debate I had with my business partner over the last 35 years. I knew him for like that amount of time, and he was really thinking that we would need to understand biology with AI, and actually, I was thinking that would be not impossible, but very difficult. And I thought that by finding some critical target and get one medication out of luck on one of the targets, like pharma is doing these days, we would be able to get an extra 15% that will give us the time to find another one that gives us another 20, and so on. But I was really betting on luck, on not luck, but this kind of like. But here, like really, I didn't think that the machine learning would actually be as precise as we have made it with being as good as the experiment. And I also did not believe that we would use anything pertaining to the quantum realm. And I see that with our docking technology, deep learning and fast computing will enable us to actually harness that information to be more precise in modulating how a compound interacts. So these are two things that are completely I mean, I had them in mind, but I gave them a very low probability of happening in that quest towards longevity. But actually, we are already harnessing their power, so it's pretty interesting.
Yes, indeed. And do you think that quantum computers will actually become usable for these kinds of applications, for machine learning applications, for drug discovery? Right now, there have been some successes in quantum computing, but at a much more rudimentary level, like making a few successful calculations. Do you think we could have a quantum computer, say, within the next decade that would actually be able to do a lot of the extensive work involved in drug discovery?
So actually, I mean, here my business partner should be the one talking about this because he's the professor in quantum information. But what we have discussed is that our docking technology will be one of the first application of the quantum computers when they will be there and really usable, because the way the information is coming and the way we will feed the neural net is really fitting. So it would be one of the first applications. So it would probably lead us to make like unthinkable scale of simulations, like like any type of molecule interacting at a much larger scale. So, and that will probably happen in the next decade. But there, I'm not. I didn't follow the latest advances, and we had lots of promises with the quantum computing. It's going to happen, but to the extent we can use it right now, I don't know, anything between 5 to 15 years, probably.
Quite interesting. And our friend John H. writes that quantum computing and controlled nuclear fusion would be profound world changers. So, technologies we hopefully all have to look forward to. And yes, within 5 to 15 years, that seems like a reasonable time frame for significant applications of quantum computing to be realized. Now, since you mentioned your business partner, there has been a bit of discussion in the chat about his original reply to you that humans would just be replaced by robots. What is his current outlook on this? Has it evolved since that time, especially since you've been collaborating together on drug discovery?
Yeah, so even though he's not the most empathic being as a as a programmer who's really into his thing, he kind of I think his objective is also to achieve like the goal of reaching this escape velocity status. He's still a little bit not as confident as I am, but I'm working on the biology side a little more. But yes, that's the reason why he's doing it. He told me, I mean, I'm not sure it's going to work. Probability are quite low because he's a very skeptical guy. But so basically, he's not developing Skynet. he's working with me to do to find medication for longevity.
Yes, indeed. And really, what matters is that people with his kinds of talents dedicate those talents to making progress. And the progress could be incremental. They could have different outlooks as to where it can ultimately take us. But as long as they're working to take us in the right direction, that's all good. So I'm glad that you were able to essentially convince him to devote his abilities to this fascinating effort. Alan also has been very generous with his support for our virtual Enlightenment Salon. He says, thank you, Dr. Weber. Keep working on the science that will benefit us all. You are making the seeds and passing them on to growers. Here's to you getting some of those fruits later. So thank you for those great words, Alan. And now I wanted to give an opportunity for each of our panelists to make either any final remarks that they want to make or ask any further questions. And we have the ability in the next 19 minutes or so to still delve into some serious discussion of any areas that people want to bring up. So who would like to start? Ben.
Neither of these questions are awesome, so I'll just ask them both, and you can choose if you want to answer either of them. First, I was curious, besides you and Insilico, if there was another very interesting player in the machine learning space for longevity medicine that you could tell us more about, just for us to have a better understanding of how the longevity field is using machine learning in general across the field. Then the other one, and I don't know how relevant this is, but I was wondering about the regulatory hurdles that you face when you are testing, or excuse me, when you are using machine learning to get the data, when you're not actually generating data that's collected on animals and then humans, but maybe you're so early in the process that it's not really a hurdle that you're even concerned about. That's more like Big Pharma and those that are getting it across the finish line, that they have to concern themselves with that. So either of those or none of those or whatever.
Okay, well, regarding competitors, like they don't really publish what they're doing so much. We've seen that Recursion is acquiring some guys who look like they are good scientists, and they are acquired Cyclica, but they're looking that they might do something interesting. There is also Atomwise. There are other companies that are Excientia as well. They have received lots of money, but I've not seen any breakthrough technology so far in any of these competitors. The most interesting thing that we have seen is AlphaFold, but it's not directly applicable to drug discovery, but we will harness its power. So that's one thing. Regarding regulatory issues, we're pretty happy about it because we're just a data company. So we get numbers and molecule structures, and we make predictions. We sell predictions. So there is no regulatory issues. We really want to focus on the science and not on the paperwork.
Yes, thank you for those answers. And I'm glad that at the level at which you're operating, there are not really significant regulatory obstacles, because indeed you are not making drugs at this stage to provide to the public. You are essentially gathering knowledge about what the potential effects of various compounds could be. And that later on could be used, for instance, to decide which drugs to do clinical trials on. But it's that clinical trial process that's ultimately regulated by agencies like the FDA. So you stated that essentially it takes 10 to 17 years to get a typical drug to market these days. And with your approach, by how long do you think that time frame could be shortened?
So, I mean, I put 10 to 17 years. There are drugs that go a little bit faster. So the idea is that, as I said, we will not focus only on drug discovery. So like next year, we should have new natural compounds that are already authorized that should be quite active. And then repurposing is taking 3 to 5 years. So these things, so we would have like very quickly some natural compounds also in a frame of maybe 2 years that they would not need the FDA things. But the 10 to 17 years, what I know is that from the 2 to 5 years, we could really squeeze that to, let's say, 3 months. That's the preclinical. After the clinical might go a little bit faster because of less back and forth, side effect cancellations, because we're really looking into many of the off-target effects, and also the profile for cytochrome inhibition. So people who usually take many drugs, they will not like make one cytochrome tired at the same time as I use two drugs that use the same enzyme. So these are things that will slightly accelerate the process. Also, COVID was pretty bad for many reasons, but it kind of enabled to accelerate the clinical side, because some of the holders were like pushed, and they fell forever. Some of them just for COVID, some of them maybe not for the best, but still it will accelerate this. So I'd hope to have like something on the market as a medication anywhere within 6, to be optimistic, to 8 years. But before, we will have some repurposed compounds and natural compounds that will be very efficient.
Yes, thank you. So quite a few years can be saved using this approach, which is quite encouraging. Daniel, let's hear your question or comment.
Yeah, I used to do a bit of public affairs radio, college radio in the 90s. And one of my favorite guests was Dr. Bob Beck, who was doctor of physics. He invented the strobe light. And he had a suite of electromedical and metallic and ozonation therapies that he claimed were capable of curing. You can't use the word cure, of course, because the AMA has a patent on that. But it was basically colloidal silver, which elemental silver has a natural antibiotic, antifungal, antiviral property. Ozonated water, which if you bubble ozone through ice water and then drink the water, you can see your pulse oxygenation rates go up from like 80 to over 100. The other was electromagnetic pulsing through kind of a coil, which you'd put over your lymph nodes. And then the last thing was a blood microvoltage pulsing biphasic DC. And apparently it has been researched at Albert Einstein Medical College, and they were doing not the in vivo version of this, where you put the little cotton wrapped electrodes on your wrist, your brachial artery and vein, but an external thing where they would actually take the blood out and micropulse it with electricity. So are these do-it-yourself kinds of medical technologies actively suppressed by the medical cartel? Why does silver have this antibiotic property? Why do these micropulsations change the cellular potential for viruses and pathogens to invade cells? Do you know of any work that's being done along these lines, or what's the promise of researching that? And can people do this stuff on themselves, experiment on themselves, and be exempt from medical regulations? So anyone can look up the Bob Beck therapy online, but it's again, it hasn't reached a huge level of acceptance, but apparently it's non-toxic and non-invasive. So the Hippocratic Oath says do no greater harm. Try the least invasive thing that might work to cure a situation, and then move on to something more invasive if that doesn't work. So any thoughts on any of that?
So I'm myself taking about 100 pills a day, and had some injuries and have done multiple treatments that were semi-experimental. So when things make sense and they are not harmful, I wouldn't say that you shouldn't try them. The zinc thing doesn't sound too bad. The ozone apparently either. Electrostimulation has shown to be beneficial in different areas. So I've not investigated it personally, rather at least not more than like for some muscle injury or like muscle stimulation. But the zinc, I used it for disinfection. It's in many ointments.
Actually, silver was the element, not zinc.
Yeah, silver. Sorry,
zinc as well. Yeah, a lot of elements have these.
Yeah, sorry, because I have like some with silver and zinc, so that's why.
Yeah.
No. There are easy things that work. I mean, like vitamin C is a good adjuvant for chemotherapy, and it should be used more, for instance. And many clinics are starting to do like injection of vitamin C. So there are basic things that can be done and that can help. And if they are not harmful, why not trying them?
But the idea is there's no money in them, so that's why they're not being advocated or adopted widely.
I mean, the silver is quite advocated, adopted, I'd say, like the vitamin C as well. But definitely, the fact that there is no patentable thing there, like is the treatment is not. Yeah, I see where you go, where you're coming from, but I think more and more now with the social media, like natural medicine and the advances of science, more and more people are experimenting on kind of healthy approaches. So I mean, if it's not harmful, why not? But then definitely these things won't be the most efficient, but as side treatments, they're definitely useful, I guess.
All right, thank you. And Art Ramon writes in the chat that he took some colloidal silver after he got COVID, but he can't say that it worked very much. So silver, as I understand it, tends to have a low toxicity, fairly low side effects, but it could be, at least in certain applications, of limited effectiveness in addressing the underlying condition as well. But let's go to Art Ramon for any closing question or comment.
Yeah, what about research into nootropics? I mean, there's a lot of supplements. I've taken a few, like racetam and some other ones that are not quite medication, but they're a compound that they make and they can sell as a supplement legally. So any interest or work in nootropics?
Yes. I mean, I have some personal interest in nootropics. I've been taking some. I also actually have some people who contacted me for psychoactive compounds that could be used for many purposes, but mostly therapeutic. I mean, that's the objective. But we have in our database some data about neurotransmitters, some receptors in the brain. So there could be a possibility of developing drug that can enhance mood and maybe also improve neuroplasticity. So far, I've just quickly, because we're discussing with some people at different levels. So here it's just like some first talks about collaboration and looking at some psychoactive drugs. I see that there is some interest, and we have enough data to find things that will be actually psychoactive. And actually, one of the things I'm taking for longevity is dopa mucuna. So it's L-dopa actually, and it looks like it extends lifespan of mice quite a lot. So definitely, probably the mood is having an effect on longevity. So the nootropic and the psychotropic must have some and some. It's kind of a fast way to access a result. So yes, we have some interest there. It's not our primary interest, but we are investigating this.
Yeah, I still deal with a lot of COVID brain fog, and I've tried various things. Some help for a bit, but it seems like they quit working after a while. It's like the body builds a tolerance to it. So, I don't want to keep living forever and dealing with brain fog, for thousands of years. So definitely want some nootropic to help me out. Thank you.
Yeah, we will be working on it, definitely.
Yes, actually researching effective treatments and hopefully cures for long COVID could be a major opportunity, because a lot of people are not only suffering from long COVID, but their suffering is quite individualized. So different people can have very different arrays of symptoms from others who are afflicted with long COVID. I also wanted to give Alan Crowley the opportunity to ask a final question or make a final comment.
Thank you. I threw my 2 cents in on the chat, and I'll yield the balance of my time to the chair. Thank you, Dr. Baber.
All right. Thank you to Alan. And I wanted to ask you, Yvonne, what do you see as the greatest challenges that might be standing in the way of the realization of your vision? What are some of the biggest obstacles that would need to be overcome in order for your approach to become widespread and to be used in drug discovery essentially throughout the industry?
It's really the low pace that the pharma industry is taking. So it's like mammoths. They have they are huge, but they have a system that's already well established, and it's difficult to make a change there. So we really have to enter into a door where they are investing into collaborations, and I see there are 3, 4 of them that are starting to build a bridge with external innovative companies, and that's towards this bridge that I think I'm going to climb there. Because directly talking to some people, I mean, at the head of chemistry somewhere, I mean, they have done things for decades the same way. And AI in drug discovery is a little bit like anti-aging. So people have been selling snake oils for thousands of years, and now that we will have something, that we have already some compounds that have some effect, people still don't really believe in them. And then with the pharma industry, they have been told, like, yeah, 20 years ago already, AI is going to disrupt drug discovery, and nothing really concrete happened until now. So they are a little bit skeptical. And there is also this problem of data exchange. But so the challenge is really to talk to the right people and that to convince them to be open-minded enough to do some tests and to change a little bit the way they are working to optimize their process. Because here we can definitely save years and hundreds of millions per drug. So it would be foolish not to move forward. So I think and I hope I'm going to be able to make that move with one of the main pharma companies soon. But it is still a challenge because chemists don't come from machine learning, and they still think that there is a little bit of black magic behind the black box. So I think we just need to educate and to really enter there and do some proof of concept in-house with them rather than external proof of concept, so they can really see how much they can save and how much time in terms of time and in terms of money.
Mm-hmm. Yes, it seems that this is an immense opportunity for those who realize it, for those who understand the significance of your approach in accelerating drug discovery and the time between the initial effort to develop an effective treatment or cure for a disease or a health condition and the time that it's actually available to patients. So thank you very much to Dr. Yvonne de Weber for joining us today and for illustrating the possibilities of machine learning. Please go to cortexdiscovery.com if you would like to learn more about this approach. I think it is quite fascinating. And thank you also to our panelists, our USTP officers, as well as Alan Crowley. Thank you to our audience for the engaging questions and comments. As always, I hope that in due time, we will benefit from this approach to discovery and in the meantime and for all time going forward, I hope that we live long and prosper.